Persons and learned audience, Assalamu alaikum and good evening. I am Dr. Shakera Ahmed, Associate Professor from Chittagong Medical College. Welcome you all to my talks on mutation spectrum and expression profiling of PAP2 gene highlights its biomarker potential for the diagnosis of breast cancer in Bangladeshi female. You know, breast cancer is a heterogeneous disease with varied molecular characteristics. Starts usually in the ductal epithelium, caused by uncontrolled growth and proliferation of cells. And genetic changes, DNA or RNA mutations, and DNA damage play the key role. Breast cancer is the most prevalent cancer of Bangladesh, and its incidence and mortality rate is also remarkably high. According to the expression of endocrine receptors, breast cancer can be broadly classified into 4 subtypes like luminal A, luminal B, HER2 enriched, and triple negative breast cancer. And you know the frequency and response to therapy. As our main focus is on PAP2 gene mutation and expression, here is a brief overview of PAP2 gene. PAP2 is the partner and localizer of BRCA2, and it's a tumor suppressor gene. It plays a key role in the homologous recombination pathway as a mediator between BRCA1 and 2 RAD51 complex. PAP2 protein collaborates with the BRCA2 protein to repair DNA damage, particularly in cases of double-strand breaks or mismatched strands. Loss of both PAP2 alleles lead to the activation of non-homologous end-joining or NHEJ pathway, resulting in genomic instability. Here is the diagram of PAP2 gene encoding chromosome, that is chromosome 16, and basic information about the PAP2, which I have already described. So germline pathogenic variants in PAP2 gene are associated with increased cancer risk, predominantly breast cancer. It's the most well-studied genes in breast cancer are BRCA1 and 2, you all know about that, but recent studies suggest that PAP2 is the third most mutated gene associated with hereditary breast cancer after BRCA1 and 2. The PAP2 protein interacts directly with BRCA1 and 2 to facilitate the repair of DNA double-strand breaks, and mutation in this gene disrupts these interactions, leading to inefficient DNA repair and genomic instability, which is a key driver of tumorigenesis. So what's the rationale of this study? Despite its clinical relevance, PAP2 remains underrepresented in genetic screening panels in Bangladesh. Moreover, overall genetic studies on the Bangladeshi population are very limited, and the prevalence of PAP2 mutation remains largely unknown. There is a notable lack of comprehensive data on the mutational spectrum and expression patterns of PAP2 in Bangladeshi breast cancer patients. Integrating PAP2 into routine genetic screening could significantly improve risk prediction, personalized treatment, and early intervention strategies for hereditary breast cancer in this region. Therefore, we investigated the mutational landscape and expression profile of the PAP2 gene in this context to contribute to more inclusive and effective approach for breast cancer management in our country. We hypothesized that the PAP2 gene is downregulated in breast cancer, more specifically in hormone-responsive subtypes, and its expression is reduced in higher tumor grades. Our general objective was to analyze the mutational and gene expression pattern of PAP2 gene in breast cancer tissue samples from Bangladeshi patients. And we set out some specific objectives according to our hypothesis. Those includes to identify and characterize germline and somatic mutations in the PAP2 gene using whole exome sequencing, to evaluate the expression of PAP2 at transcriptional level in breast tumor tissues compared to adjacent normal tissues, to explore possible correlations between PAP2 alterations and clinical parameters such as tumor subtypes and grade, to assess the potential of PAP2 as a biomarker and its implications for personalized therapy in the Bangladeshi breast cancer population. Let's come to the methodology. Total 30 patients were included in this study, and we have collected the tumorous tissue and the adjacent non-tumorous tissue of those patients during their surgery. And we have confirmed the breast cancer by histopathology, and the subtype was determined by immunohistochemistry. To detect the mutation of the PAP2 gene, we have extracted total DNA by using commercially available kit, and then the DNA quantity and quality was assessed using nanodrop spectrophotometer and agarose gel electrophoresis respectively. Then we have done the library preparation for sequencing, and finally the next generation sequencing was done on Illumina platform. And then for PAP2 gene expression, we have isolated the RNA, and RNA quality and quantity was also determined. And then complementary DNA synthesis was performed, and finally quantitative RT-PCR was done to assess the gene expression. Now come to the results. We have done the whole exome sequencing of 10 breast cancer patients after extracting the DNA from those patients' tumor cells. And analysis revealed that the mutational was detected in the position of chromosome 16 in the position of 2362946, and G2A substitution was observed mostly in the luminal and TNBC subtype of breast cancer. In HER2 positive breast cancer, mutation was also detected in the chromosome 16, but it was in the different position. Then quantitative RT-PCR was done to see the expression of PAP2 gene in breast cancer, and it has been seen that expression of PAP2 is significantly downregulated in breast cancer tissues in relation to the normal adjacent tissues. According to subtype, it has been seen that expression of PAP2 is significantly downregulated in luminal and TNBC subtypes. And according to tumor grade, it was significantly downregulated to tumor grade 2 and 3. Expression pattern of PAP2 gene was also seen in the TCGA dataset, and expression of PAP2 was significantly upregulated in primary tumor, which is discordant with our findings. Expression pattern according to subtype in TCGA dataset also shown that it was significantly upregulated in luminal and triple-negative breast cancer. Expression pattern according to the stage, it has been shown that expression was significantly elevated with the advancement of stages. Finally, the overall survival was seen in the Kaplan-Meier plotter tool, and it has been shown that low expression of PAP2 is associated with poor overall survival. So in summary, PAP2 mutations are mostly reported at chromosome 16, 2362946 position, where G was substituted to A in luminal subtype. Expression of PAP2 is significantly downregulated in breast cancer tissues. It was significantly downregulated in luminal and TNBC subtypes, but in expression in the and also downregulated in tumor grade 2 and 3, but low expression of PAP2 is associated with poor overall survival. Expression of PAP2 is shown significant upregulation in primary tumor in TCGA dataset, and it was shown significant upregulation in luminal and TNBC in TCGA dataset. So in conclusion, we can say that PAP2 may serve as a promising biomarker for breast cancer diagnosis in Bangladesh and could be included in gene panel to improve breast cancer management. I like to acknowledge my collaborator, Dr. Mohammad Mosharraf Hossain, Department of Biochemistry and Molecular Biology, Chittagong University, and his lab members, and also Chittagong Medical University for financial help. So thank you so much for your patient hearing.